The research paper we reviewed is called The Comparative Efficacy of Feline Leukemia Virus, FeLV, Inactivated Whole Virus Vaccine and Canarypox Virus Vectored Vaccine during Virulent FeLV Challenge and Immunosuppression. This was done by Denise Andriotti and myself, Skylar Adams.
Feline leukemia virus is a gamma retrovirus that is distributed worldwide. It was first discovered in 1964. The first diagnostic test was not available until 1973 and the first vaccine in 1986. Infection occurs among domestic and wild cats to include lions. Transmission occurs vertically from infected mother cat to kittens, or horizontally among cats who share litter boxes, feeding dishes, from mutual grooming, or from a bite. The virus is shed in very high quantities in saliva, nasal secretions, feces, milk, and urine. Risk factors include young age, high population density, and poor hygiene. There are three main subtypes of feline leukemia virus. Subtype A is involved in every infection. Subtype B originates from recombination of feline leukemia virus A and envelope feline leukemia virus. Subtype C results from mutations in the envelope gene. Subtype T has a tropism for T lymphocytes. There are many clinical signs of infection which include cytoproliferative disorders, cytosuppressive disorders, inflammatory disorders, abortions, enteritis, and more. There currently is no cure for feline leukemia virus, and it is usually fatal. The average survival after diagnosis is 2.4 years. There are 6 stages of the virus. In stage 1, the virus enters the cat and infection begins in the oropharynx, where the virus infects epithelial cells, B lymphocytes, and macrophages. In stage 2, the virus enters the bloodstream and continues to spread throughout the body. In stage 3, the virus enters the lymphoid system and spreads further throughout the cat's body. Stage 4 is the main point of infection. If the cat's immune response is ineffective, the virus overcomes the cat's immune system and causes viremia. In stage 5, the bone marrow is infected and the virus stays with the cat for the rest of its life. Lastly, in stage 6, the cat is overwhelmed by infection. The mucus and glandular epithelial cells become infected. Now the virus can easily be transferred to other felines. There are 3 possible outcomes after infection. In regressive infection, the cat's immune response is able to clear the virus and the cat becomes resistant to future infection. In progressive infection, the cat may fail to mount an effective immune response and becomes persistently viremic with no clinical signs. In active infection, the cat shows clinical signs and secondary viremia. The outcome depends on the cat's age, immunocompetence, concurrent disease, viral strain, dose, and duration of exposure.
There are 4 feline leukemia vaccines available in the United States commercially. 2 whole virus adjuvanted killed vaccines are available. 1 dual adjuvanted multiple antigen vaccines available, and 1 non-adjuvanted canarypox virus vectored vaccine is also available. The Novavax Feline 2 FeLV vaccine and the PureVax recombinant FeLV vaccine are the 2 that this research project studies. And the Novavax vaccine is one of the 2 whole virus adjuvanted killed vaccines, while the PureVax vaccine is the only non-adjuvanted FeLV vaccine available, and it's a canarypox virus vectored vaccine. The Novavax vaccine and the PureVax vaccine are both recommended to be given to a cat whenever it is 9 weeks old, and then it is recommended to be given again as a booster in 3 to 4 weeks after that. The Novavax vaccine is then recommended to be given biannually or every 2 years after that, while the PureVax vaccine is recommended to be given every year after that. The Novavax vaccine has been studied and challenged more than the PureVax vaccine has, and those studies have revealed that the prevented infection ranges from 90 to 100%. The PureVax vaccine, though it has limited research done on it, has been shown to have a prevented fraction of 20% in a previous study. There are no sterilizing FeLV vaccines. The FeLV vaccines are meant to prevent active or progressive infection from occurring and attempt to maintain a regressive infection so that it is more manageable and sometimes can even cause the host to be able to get rid of the infection altogether. This study was designed to compare the efficacy of the whole virus adjuvanted killed vaccine, the Novavax vaccine, and the PureVax non-adjuvanted recombinant FeLV vaccine, and to just gather more information about the efficacy of the PureVax vaccine because there has not been as much studying done on it. The test subjects were 32 8-week-old kittens that all tested negative for the P27 capsid protein via ELISA. They were divided into 3 groups, a Novavax group, a PureVax vaccine group, and a placebo control group. They were vaccinated at 9 weeks of age, and then they were boosted again at 12 weeks. The FeLV subtype A was found in both vaccines and in the virus that they were challenged with, but it was considered a heterochallenge after analyzing the alignment and amino acid sequence because they were different enough. They were considered different viruses. The subjects were immunosuppressed at least 4 hours before they were challenged, and then again a week after they were challenged. They were tested for IgG antibodies with the ELISA at 20 days and at 111 days. They were tested for the P27 antigen with ELISA every week from 3 to 9 weeks after challenge, and the viral RNA was tested with real-time reverse transcription PCR every week from 3 to 9 weeks, and the FeLV DNA was tested for with real-time PCR at every week from 3 to 9 weeks as well.
After a vaccine is administered, the immune system recognizes the antigens it contains as invaders. T and B lymphocytes are activated, leading to the development of antibodies. Neutralization of pathogens at mucosal surfaces is achieved by vaccine-induced serum IgG antibodies. The figure to the left shows serum IgG antibodies to feline leukemia virus vaccination. Prior to vaccination on study day 0, all cats were negative for feline leukemia virus antibody. On study days 20 and 111, 10 of the 11 cats in the Nobivac group were positive for feline leukemia virus antibody. All cats in the PureVax recombinant feline leukemia virus group as well as the control group remained negative for the feline leukemia virus antibody through the entire vaccination phase. The figure to the right shows the P27 antigen, proviral DNA, and the viral RNA results by weeks post-challenge. Cats in the Nobivac group were negative for P27 antigen throughout the post-challenge period. 50% of the PureVax vaccinated cats and 91% of the control group were positive for P27 antigenemia. P27 is a core protein of feline leukemia virus that is produced in abundance in infected cells. The presence of P27 antigen indicates that infection is present. At 9 weeks post-challenge, plasma viral RNA was detected in 1 of 11, 9% of the Nobivac vaccinated cats, in 6 of 10, 60% of PureVax vaccinated cats, and in 9 of 11, 82% of the control cats. From weeks 7 to 9, viral RNA loads were significantly lower in the Nobivac group than in the PureVax group. The detection of viral RNA can be taken as a marker for viremia. Proviral DNA was detected in 1 of 11, 9% of the Nobivac vaccinated cats, in 6 of 10, 60% of the PureVax vaccinated cats, and in 9 of 11, 82% of the control cats. From weeks 6 to 9, cats in the Novivac group had significantly lower proviral DNA loads than those in the PureVax group. As a retrovirus, feline leukemia virus uses its reverse transcriptase enzyme to create a DNA copy of its RNA viral genome. This proviral DNA copy is then inserted into the genomic DNA of the infected feline cell when it divides. As long as that cell survives, the feline leukemia virus proviral DNA persists and leaves behind a blueprint for infectious virus. The presence of proviral DNA confirms the presence of feline leukemia virus infections that have progressed to the bone marrow. There are several critiques regarding this study. First, there needs to be a laboratory challenge model that closely mimics natural infection. For instance, in this study, cats were given MPA the day of challenge and then another equally high dose a week after challenge in order to induce immunosuppression. This is not the situation most often encountered in the field and can basically create more favorable conditions for inactivated vaccine efficacy. There also wasn't a large sample size in the study. Third, the study used young male cats that were castrated. It was never indicated that gender was a factor in randomization, so the study should have introduced like an equal number of cats from each gender. And lastly, it would have been better if there were no adverse events involved in the study. 3 cats were euthanized due to adverse reactions to MPA, and 1 cat did not recover from anesthesia.
These are our sources. The top one is the research paper that we reviewed, and the rest of them are supplemental resources that were used.